Hello everyone, my name is Ryan Green and today I will be presenting my research project, which is the optimization of an in vitro model of neuroinflammation exposing Shy-Y neuronal cells to stimulated THP1 conditioned media. To begin with, neuroinflammation is an immune response within the central nervous system, and it's commonly involved in the progression of many neurodegenerative diseases like Alzheimer's and Parkinson's. These can happen when immune cells are overactive and release excess pro-inflammatory cytokines like TNF-alpha, which can lead to neuronal damage or neuronal death. Previous studies have shown the potential of a stimulated THP1 media treatment on Shy-Y cells as a model for neuroinflammation. So the objective for this study was to replicate this and to further it and optimize it. So using THP1 monocytes, stimulating them for an immune response, and then treating Shy-Y cells as a representative of neuronal cells. It was hypothesized that the Shy-Y cells exposed to this stimulated THP1 conditioned media would provide a successful model of neuroinflammation. And the goals from this study were to both validate and optimize that model, to compare it against a standard drug like prednisone, and also to measure levels of TNF-alpha produced by different methods of this model. In terms of methods, it was culturing both cells, including the Shy-Ys and THP1s. The THP1s were then differentiated with 200 nanomolar PMA and stimulated with 1 microgram per mil LPS. That conditioned media that was taken from these cells was then used both as a treatment on Shy-Y cells for a cell viability assay and also to measure the analyte for TNF-alpha concentrations. This was the basis for the study, and it also involved many different changes to the model to measure the outcomes in terms of cell viability and TNF-alpha concentration, including the length of treatment, stimulated versus unstimulated THP1s, and the difference between different cell densities, and finally testing with prednisones to observe the difference in levels from the model when it's treated with an anti-inflammatory drug. In terms of results, after 48 hours, there was no significant change in any of the dilutions of conditioned media in terms of cell viability. Meanwhile, after 72-hour exposure, the neat conditioned media had a significant decrease in cell viability compared to the positive or the negative control, sorry. This showed both that the 72-hour exposure was ideal for a cytotoxic effect and also using undiluted media was necessary for the model. For cell density, the 2 times 10 to the 5 cells per mil THP1 media did not show any cytotoxic effect, so higher cell densities were tested. At 4, 5, and 6 times 10 to the 5 cells per mil, there was a significant decrease in cell viability compared to the negative control and compared to the unstimulated levels at those same densities. This was also reflected in the TNF-alpha concentration levels from the ELISA, which showed very high levels in those stimulated samples. In terms of prednisone treatment, the higher prednisone treatments showed no change in cell viability, which was an improvement compared to the untreated models, which showed a decrease. This showed the potential for an anti-inflammatory effect, which was represented in the model, and it's also reflected in the ELISA data, which shows an increased TNF-alpha levels at lower prednisone concentrations. Overall, this study successfully established and optimized an in vitro model of neuroinflammation using these THP1 media and Shy-Y cells, and it leaves plenty of room for growth. So far, it's determined that cell density of 5 times 10 to the 5 cells per mil, 200 nanomolar PMA, 1 microgram per mil LPS, and a 3-day treatment period are ideal. It also showed an effect when treated with prednisone, which shows its potential as a model for tests of any other anti-inflammatory drugs. In terms of future considerations, they could mainly replicate the current findings, since certain MTT data and ELISA data did not have many replicates and could be furthered to determine any more significant difference. But it could also test different ranges of prednisone or other neuroprotective compounds. It could differentiate the Shy-Y cells to provide a more accurate neuronal cell environment. And it could also use different different cell assays like LDH to show cell viability, or even use qPCR to incorporate gene expression to better understand the model.